Good afternoon, everyone. Welcome. I am Jerry Bowers, and I'm the VP of External Affairs here at the lab, and we are very excited to have you all here to learn about the most exciting topic of the summer, fruit flies. Who doesn't love a good fruit fly? I'm sure many of you, like me, probably are intimately familiar with fruit flies this time of year. It does seem to be the season. But our goal today is to have you leave here with an entirely new image of the fruit fly. So the next time you swat one away when you're in the kitchen, we hope you're going to really think about what amazing scientific warriors they are, and actually really powerful tools in scientific research. So tonight we have Dr. Liz Marnick, who's going to moderate our discussion with Dr. Travis Carney, and they are going to tell you a little bit about the research that's happening here at the MDI Biolab on fruit flies, but also just about the enormous role that fruit flies play in biomedical research around the world. So with that, I'm going to turn it over to you guys. I do want to just quickly remind the folks on Zoom that this is Q&A. Like, we want this to be very interactive. So for those on Zoom, feel free to use the raise hand feature if you want to participate, or just raise your hand, turn your camera on and raise your hand. We'll be monitoring Zoom and monitoring the chat, and hope that we have a great discussion. So thank you all.
All right, before we jump into talking about fruit flies, I was going to have Travis give us a brief introduction to him, because I know a lot of you probably haven't gotten to meet him in different settings yet.
Hi, I'm Travis. My name is Travis Carney. I'm originally from the Pacific Northwest. Raise your hands, Pacific Northwest. Yes, fantastic. Yeah. Great. That's so I was raised in northern Idaho, and I was a very, very small town boy. My hometown was like 50 people, not 50,000, but 50. And then I went to Western Washington University in Bellingham, which is right up near the Canadian border, for my undergrad. And got a little bit of undergrad research experience there. We were doing microbial ecology, which was a lot of fun and very interesting, but it turned out not to be my cup of tea. I did a one-year post-bac. This was a research program that was designed for people who had just gotten their bachelor's degree. That was in Los Alamos, New Mexico, at the national lab there. And they don't only do nuclear research. There's a small biological division there as well, and they do national defense stuff. So that was also microbial ecology work. And it was there that I decided I wanted to go on to graduate school. So from New Mexico, I went back to the Pacific Northwest. I went to the University of Oregon for my PhD. Yes, thank you. We are the Ducks, that's right. And I ended up joining a fly lab. I kind of fell in love with a project that one of the graduate students was doing there. I rotated in their lab and decided to research fruit flies. And for my PhD work, I was looking at neural stem cells in the fruit fly, which, full circle, that's what I'm looking at again today, that I'll talk to you about in a little while. And a cool thing about working with fruit flies is that if you end up working on a gene that nobody's really studied very much before, if it doesn't have a name, you get to name the gene. And so for my PhD work, I got to name a fruit fly gene, and I named it midlife crisis, which was apropos of nothing, I promise. No, of course, it has to do with the phenotype that I was studying in neural stem cells and in neurons. So I completed my PhD, and my wife and I decided that we'd kind of gotten the travel bug, and we decided that we wanted to go abroad for a postdoc. And so instead of looking in the US, I looked in Europe, which is a little bit unusual, but there's a great deal of great research going on in Europe as well. So I got a postdoc first in Switzerland, and it was only a short time that I spent there, about a year, and that job ran out of funding. And just about the time I fell in love with Switzerland, I had to leave Switzerland. But I was fortunate to get another postdoc in Germany with Professor Helena Shcherbata, who had a lab at the time at the Max Planck Institute. And So I joined her there. That was in Göttingen, Germany. And then our lab moved to Hanover Medical School in Hanover, Germany. And that's where her lab is now. So I had a postdoc with her for several years. And then my postdoc ended and I went back to the Pacific Northwest. And a few months later, this job at MDI came up. And I jumped on the possibility of coming here and starting up a fruit fly lab, which is a satellite lab of Professor Shcherbata. So she's got her lab still in Germany, and I'm here running the fruit fly lab in her absence. She's here part of the year, but I'm here full time. And yeah, and when I was in Germany, I studied several different systems, including the fruit fly testes, which is a really cool system that I'm not going to talk about today. But since I'm back here, since I'm here in Maine now, our lab is studying the fruit fly brain, which is the full circle that I discussed before.
So I'm going to ask you guys some questions, you guys being the audience, a few questions throughout the next hour. And Jerry kind of already hinted at this, but I want to see raise of hands. How many of you have squashed some fruit flies in your house thinking that they were very annoying? Raise your hand. Most of you probably have at some point. My husband actually hates fruit flies with a passion, and anytime I use them for outreach, he threatens to divorce me if I bring them home, which they don't escape, they're fine, but he's not a fan of fruit flies. I hope by the end of this, when I ask you that question again, whether or not you think fruit flies are annoying, you have a different perspective, but we're going to wait and see. So Travis, can you give us a little bit of information about why fruit flies are such a good tool for scientific research?
Yeah, absolutely. Roy, can I have the slides or do I need to do it from down here?
There you go.
Fantastic. Well, there's the title slide. This is a fruit fly brain from the larva. And there are a whole bunch of reasons that fruit flies are a great system to study, and I'm just going to highlight a few of them. So up here, I've got the fruit fly life cycle. And as you can see, the life cycle is only 10 days, which is quite rapid, much more rapid than a vertebrate system would be. Not as fast as worms, but still rather rapid. And the life cycle is the time from when an egg is laid until it's gone full cycle to an adult. The adult itself can last up to 2 or 3 months. And so that means that not only can we do rapid research by looking at these, either the larvae, or a lot of research has been done actually in the embryo during this life cycle, but it also gives us the ability to do aging research by looking at 2 or 3 month old flies with a mutation of interest or something like that. So the rapid cycle is something that we really love. My PhD work was done looking at the brains of the third instar larval stage. This occurs about 5 days after the embryos are laid. And I would dissect the brains out of them and look at the brains for different phenotypes. And then in my postdoc, I started looking at adults, and you have to wait an entire 10 days for the adults. So this seemed incredibly slow. So I'm very happy to be looking. I'm happy when I can look at larval stages again. So flies are insects, and so they go through this life cycle where the larvae that I mentioned grow and go through several stages, and then they pupariate. They form pupae. And this is the same as a cocoon of a butterfly, where it forms a cocoon and it stops moving for a period of time. And in that pupal case, it undergoes metamorphosis, and the body plan changes drastically, and out comes the adult fly. Another really important reason that we love working on flies is the simplicity of their genome. So humans have 23 pairs of chromosomes, which equates to about 3.1 billion bases. That's the letters of the code of DNA. And flies are much, much smaller, with about 160, 165 million base pairs, and only 4 chromosomes. And I'll point out that that 4th chromosome you might be able to barely see here is very, very small. And we almost never use the 4th chromosome. It's so small, and there's really one important gene on it, an I gene. And so most of the time we neglect the fourth chromosome. We don't need it. And so really it's only those 3 chromosomes that we're worried about. And so that allows us to, it makes it a lot easier to do genetic crosses when we don't have to track as many chromosomes as one would have to do in a vertebrate system. In flies, we have a huge number of genetic tools that we're able to use. And this is because fruit flies have been used for over 100 years now as a model system. There was a researcher, Thomas Hunt Morgan, who started working on fruit flies in 1910 at Columbia University. And he was first interested in it for evolution reasons, but then they found a mutant fly that had white eyes. Most flies have a beautiful red eye, but this mutant had a white eye. And they followed up the genetic cause of what was causing the eye to be white, and they realized that they'd be able to use these flies as a genetic organism to study how genes are passed on and how they interact with each other. So that was over 100 years now. And fruit fly research has gone on continuously since then. And now we have the ability to use many of the tools that have been generated by fruit fly researchers over all of those decades. And when I say tools, I mean a piece of DNA that somebody made artificially in the lab, and then they put that DNA into the genome of the flies, and that can do something that's interesting or useful to us. We can use that to either turn on genes when we want to, or turn the gene level way down in whatever cell types we want, or it can tell us something interesting about the gene itself, like where, in which cells a particular gene is expressed. Is it in neurons, or is it in the gut, or things like that? And so those tools have been passed down through the generation until we today can take advantage of those. And I just have this on here. This picture is taken from the Bloomington Stock Center in Indiana. This is the largest stock center in the world. And they house tens of thousands of different Drosophila stocks. Each stock has a different mutation or a different tool, like I just mentioned, that help us to determine what genes are doing. And they maintain those stocks generation after generation, year after year. And then if we want to study one of the stocks that they have, all we have to do is order it from Bloomington. And it arrives in a week or two, and then we can study it. And so among the stocks that they have is the mutation in just about every gene in the entire genome. So if you are interested in a process that's going on in a cell, and you find yourself interested in a particular gene, there's a very good chance that there's already a mutation in that gene available, and you can just get it delivered to your door and study that. So it's impossible to overstate the value of a stock center like this and the collection that they have.
You can almost think of it like a library for fruit flies.
It's fantastic, yeah.
So you might be getting into this a little bit later, but when you talked about the chromosomes, how much of the genes you find in a fruit fly are the same as the genes you would find in a human? What's that similarity like?
So about 60% of the genes that fruit flies have we also have in humans. But even more remarkable than that is that if you just look at the genes in humans that are known to cause some disease, it's more like 75%. And so the similarity, when you're just looking at those genes that are really important and are known to cause a disorder, it's 3/4 of the genes. So if there's a disorder, a disease that you want to study and its genetic cause is known, then you can study that in flies.
Yeah, and that's a common trait you'll notice with a lot of the model organisms we use here at the lab, in that there are those genes that are, even though a fly or a worm looks so different from a human, there are those genes that are the same and do similar things. So by studying them in an organism like the fruit fly, we can learn more about how they may also work in a human organism. So now, a lot of you have said you've seen fruit flies in kitchens, but they probably have flown away from you. So we are going to circulate some fruit fly vials. Don't uncork them, please. But I'm going to pass around some of those vials so you can take a little bit closer of a look at what those fruit flies look like without them flying away from you. And please go ahead, pass them around to everyone. We want to make sure everyone gets a chance to see.
So these first two vials, the green tag is a wild type, that's a normal fly, and the red one is a mutant. And how disappointing. And then the rest of these are,
Thank you.
you're welcome.
I did my PhD thesis on larval salivary.
So you've seen fruit flies is what you're saying?
So can you say again what they're looking at with those different
Yes.
vials that are being passed around?
In the vials that have adults, one of the vials is wild type flies. Those are normal flies, completely normal. And the other one with the red tag is a mutant. And that particular mutant causes the flies to have curly wings. And so if you take the adults and just hold them up to the light, I hope that you'll be able to see that one of them has perfectly flat wings. I should go to the next. I don't have my clicker. One of the flies will have perfectly flat wings, but the mutant flies will have curled wings like this. And then the vials that don't have adults in them, what they have in them is larvae and pupae. And so you'll see a lot of pupae just on the side of the vial. And then in some of those vials, perhaps all of them, you'll also see larvae, these third instar larvae, the largest ones that are crawling up the side. The larvae will be white, so and should be moving as well. So you'll be able to recognize that those ones are still larvae. And what they're looking for a place to form pupae. And one thing about the larvae and pupae, if you look carefully, you'll see two different sizes in there. The wild type pupae are long like this. That's a normal pupa. And then some of them are what we call tubby, and they're about half that length. They're short and fat.
Fruit fly has the best, like you guys have the best mutation names.
Yeah. Yeah, it's fun sometimes.
I think we can take, if anyone has a question about what they're looking at. Anybody? Yeah.
Did you say you were studying neuroplasticity or neural pathways?
We are going to get into the actual research he's studying in a second. So hang tight for that question, and if we don't answer it again, I'll come back to you. So for now, if anyone has a question about the flies you're looking at in the vials, let us know. If not, you guys can keep passing those around while we'll start talking about some other things. But any questions online so far? Awesome. Okay, so then my next question to you, Travis,
Yes.
is that you talked a little bit about how much fruit flies have contributed to research over the years. And I know that they've contributed a lot of Nobel Prizes. Is there one example that you could share with the audience about how that work contributed to a Nobel Prize?
Yeah. I mentioned before the researcher Thomas Hunt Morgan. He was the first person to start studying fruit flies and their genetics. And he did end up winning a Nobel Prize, but that's not the one that I'm going to illustrate or talk more about. He won the first Drosophila Nobel Prize in 1933. But one of his students is the one that I wanted to highlight, and his name was Hermann Muller. And he did his PhD with Thomas Hunt Morgan there in New York City at Columbia. And he was very interested in mutations. And he was very interested in how mutations occur. All of what they were looking at the time were spontaneous mutations, just things that happen naturally in the fly, like the white eyes that I mentioned. This was just something that naturally happened, and they happened to find it. But he wanted to know whether this is something that could be harnessed, and whether mutations were something that we could make in the organism. And so he studied a lot of chemicals and treated the flies with chemicals, and then and followed them to see whether that caused mutations to occur, and had not a lot of luck with that. But what he did find was that X-ray irradiation causes mutations. And this is well known now, but they didn't know this at the time. There were people who used radiation in their research and didn't know that it was something that was harmful. But when he found that when he irradiated flies, it would not only would it cause mutations to occur, but also it would rearrange the genes on a chromosome. And he followed that up, and then found out how those mutations were passed on to their progeny. And I think and then he ended up winning the Nobel Prize in 1946 for showing that X-ray irradiation can cause mutations in DNA. And this is very important for what we do now, because much of the research that went on in the intervening decades between his discovery and present day was using mutations that researchers generated in genomes, not only of flies, but of other organisms as well. And then finding out what gene was mutated and finding out what that gene did in the cells. So being able to mutagenize, cause changes in the DNA was groundbreaking. And then one of his students actually found out that you could in fact use chemicals. There are chemicals that will also break DNA and cause mutations. And that was mustard gas. And she discovered that during the Second World War. And we don't use mustard gas in the lab right now, but there are other chemicals that still to this day, there's one EMS that people still use to intentionally cause mutations in animals, or animals like Drosophila, and then follow that up and see what those mutations have done to the cells that they're interested in, or the genes that they're interested in. So I think that's a really important one.
And that's a cool one, because if you think about it, right, if you don't know how something works, one of the things you can do to try to figure that out is break different pieces and see what happens. And that's essentially what is happening by doing these types of mutagenesis screens that we now know, thanks to these scientists, is that we can break a gene and then see what happens. Maybe the fruit fly dies, and then we know, well, that was really important for life, and we have to go somewhere else. Or maybe it causes a weird appearance in the fruit fly, and then we can study that. But by breaking those genes, causing those mutations, that can help us learn more about how they work. And again, because of that similarity, that also gives us insight in how those genes might be working in a human.
Cool. Just a fun fact for the audience. So another one of Thomas Hunt Morgan's students was a woman named Margaret Lewis. And Margaret Lewis was an early scientist here at the MDI Biolab. And Thomas Hunt Morgan came to the cell carcinoid lab, and I think around 1916, and spent the summer doing research at MDIville with Margaret and Warren Lewis. I love
Fantastic.
that. Full circle.
Did he bring fruit flies to the lab?
I don't know.
So the fruit flies with the curly wings, is that a naturally occurring thing, or is that something that you used some intervention?
This is it's not a naturally occurring mutation. This is a mutation that somebody intentionally, they made mutations in the fly and then isolated a fly that had these curly wings. And we use it as a marker in our flies. And we put this, so flies have two of every chromosome, just like we do. They get one from mom and one from dad. And when we have this curly mutation, we can put it on the opposite chromosome against a mutation that we're interested in, and then we can maintain the flies in such a way. So then we know when we look at the fly, if it's got the curly wings, then it has one copy of the mutation we're interested in on the opposite chromosome.
And you do that with a lot of different markers, right? Eye color, whisker length, like all these different ways you
Yeah,
have.
that's right. So I didn't go into this much, but that's what the purpose of this slide was actually. So the curly wing is a great example of one of these dominant markers that we use to look at flies. But just like Jerry said, we look at the eye shape. This one's called drop because the eyes are shaped like a drop. And then we can look at the bristles, either the pattern or the shape or the size of the bristles that cover the entire adult fly's body. You can see all of these little hairs. And does anybody see the difference in the bristle pattern besides Ian? Between this wild type fly and this one that's called humoral. So there's a difference, and when you're studying flies, you get really good at spotting it. But what's amazing to me is that somebody, probably in Thomas Hunt Morgan's lab, was just looking at flies looking for things like this, and they found it. So the difference is in these bristles right here, it's kind of right behind the eye, right on the very front part of the thorax. And this fly has 1, 2, 3 of these big dark bristles, and this fly has more. And so we use this in the same way that we use curly, although it's on a different chromosome. But if we see this marker, then we know something about the genotype of the fly. And then these are more bristle markers. This is what wild type bristles look like. This one is called singed, and I hope you can see from back there that they're kind of curly, like the fly has been singed by heat. This one we use very frequently. It's called stubble, and all of the bristles are just shorter than they are in wild type. And then scutoid. I do not like scutoid, but it exists. So I don't use it if I can help it. But the fact is that this little part of the fly right here is called the scutellum. In a normal fly, there are exactly 4 bristles on that. There's 1, 2, 3, 4. And in this genotype, there are fewer. This one, they gave a really perfect picture where all 4 are missing, but it's not always so clear as that. That's why I don't like it.
All right, we're going to start getting into more of the research that Travis actually does here at MDIBL. But first, I want to ask you guys a question. So how many of you know somebody who has a neurological condition, maybe epilepsy or ALS or autism or Alzheimer's? If you know somebody with a neurological condition, raise your hand. So pretty much everyone's hand is raised. So what you're going to learn is that we can actually use fruit flies to better understand those diseases. And Travis, can you give us a little taste of why that's the case?
Yeah. Not only do we have a lot of the same genes as fruit flies, but a lot of the cellular processes that happen in fruit flies are very similar to how they happen in ours, and that includes in neurons. And so the way a neuron works, how a neuron fires and how it communicates with another neuron is very similar in flies as it is in humans. And so we can use the fruit fly brains to model this. So this is what we're focusing on in our lab here is the fruit fly brain. This is a picture of a larva that's blown up, maybe one of these larvae, and you can see the brain is just up here near the head, and the brain is composed of 2 brain lobes like this, and then this ventral nerve cord that is something similar to your spinal cord. And in the larval brain, there are these neural stem cells, and they're shown here with the large circles. There's the green circles here, and then these little purple ones. And these neural stem cells divide over and over and over, and with each division, they make a neuron. Actually, they make 2, but they're making lots and lots of neurons, and they do this throughout larval life. And so each of these stem cells has the ability to make hundreds of neurons. And this is a picture of what that looks like. This magenta cell here is a single neuron, and then this whole cluster of blue cells are a bunch of neurons that were generated from that neural stem cell. And then those neurons, they go through processes where they mature, and they send out processes that then connect with another neuron somewhere in the brain. And not only that, oops, oh no. See, I knew I was going to do that. Wrong button. All of the neurons that are formed during larval life are the neurons that form the brain of the adult. So they go through all these maturation processes and they find their right partners and they form structures. And that is what the adult brain is composed of. And here I'm showing another larval brain. That's just the brain lobes from here. And this is a whole bunch of these lineages. So it's the stem cells and all the neurons they've generated. And in green is one kind of neural stem cell that's called the type 2 neural stem cells. And then all the magenta cells are the type 1s. And there are important differences between those that we study. In the adult brain, those neurons have matured and they form structures. And a couple examples of those are this L-shaped structure, which is called the mushroom body, and that's a learning center in the brain. These big balls, those are called antennal lobes, and that's the part of the brain that receives smells from the outside. And then this is just another fruit fly adult brain, and there are some other structures in here. These structures here that are colored, it's called the central complex, and it's required for the navigation of the fruit fly when it's flying or walking. Yeah. Do we use rapamycin? No, not currently.
So to just reiterate,
Yeah.
the fruit flies are really great organisms for studying these types of neurological conditions because the brain processes and the neurons are very similar to what we see in humans.
Yes, and that's part of it for sure. And then another huge part is that due to these very stereotyped structures that are formed in the brain, flies have behaviors that are also very stereotyped. And flies, you wouldn't think that they have complex behaviors, but they certainly do. So they obviously they walk and they fly, but also they climb. Flies tend to want to climb up, and they also climb toward light. And that's very similar in adult flies. And so that's something that we can see if there's a defect. And then they also have memory. They have the ability to remember and the ability to forget. And so I mentioned that this structure, it's called the mushroom body, and it is the learning center of the brain. And since these behaviors are very stereotypical from fly to fly, and we can look at many, many flies, we can look at and see if there's a difference in a behavior between a normal wild type fly and a fly with a mutation that may be similar to a mutation that we're interested in that humans also have.
Yeah.
How do you test whether a fly is live on its memory?
Yeah, that's a great question. So the question is, how do we know if a fly has lost its memory or its ability to form memories? And it's a simple test, but it's kind of elegant in its simplicity. And so you put flies in a little maze, a very simple maze, where they go in one tunnel or a tube, and then it forks, and then there are two branches, and you have on the ends of those branches a couple of different smells. And you can do this in two different ways. You can either give them a reward that's associated with one of those smells, or you can give them a small electric shock punishment that's associated with the smell. And it turns out that when flies do this a few times, they will learn to associate a particular smell with either good or bad. And then if they have the ability to form these memories, then you'll find that they more frequently go to the one that is more beneficial to them. And then of course, for forgetting, you can age those flies a little bit and put them through the same test and see if they can still remember which is the better tunnel to take. Yes.
They have amyloid beta and tau proteins in the brain.
Yes, they do. They have amyloid beta and tau, and there's quite a bit of active research in fruit flies and in looking at both of these. And not only that, I didn't mention this before, but we have the ability to put human genes into a fly as well. And especially with the advent of CRISPR technology, where we can make very targeted mutations and we can add genes to the fly's genome, people are looking at what happens if a human tau protein, for instance, is expressed in the brain.
Yeah, why do we care about amyloid and tau?
These are proteins that are associated with certain neurological neurodegenerative disorders. So if there's too much of these, or if the proteins themselves are dysregulated, if they're defective in some way, then they can bunch up and form aggregates of protein, and that can be very damaging to a neuron.
So I have some questions for you guys now. I'm going to test how well you remember some of the things that Travis has told you about. So if you can go to the next slide. So which one of these is a lie? So I'm going to give you a second to read these. I want you to raise your right hand if you think the lie is that fruit flies have over half the same genes as a human. Raise your left hand if you think it's a fruit fly's brain that has more than 1 million nerve cells. Or raise both hands if you think the lie is that studying fruit flies can teach us about autism and other brain disorders. Well, which one is the lie?
Oh,
Which one is the lie?
Oh.
I know. You're not getting graded, so don't worry.
There's a lot of giggling going on right now.
Giggling in that back corner. I see you guys.
Support online people are like, how do I raise a left?
Oh, I know. Sorry, I didn't think about Zoom when I made that. My fault. You can type A, B, or C in order. So A is the first, B is the second, C is the third, if you would like to contribute. All right, go to the next slide. So the lie in this case is that a fly's brain has more than 1 million nerve cells. What's the amount approximately?
They have about 200,000.
So
nerve
still
cells.
a lot.
A lot.
But not quite 1 million. And that was a hard one. But hopefully made a little bit easier that we did talk about the first one and the last one. All right, so now can you go into a little bit more details about what your research here is looking at? So we know a little bit more about why it's important to use a fruit fly, but what is it specifically that you're looking at?
Okay. In our lab, we're studying a gene called RBFOX1. And RBFOX1 it's a gene that fruit flies have. That's how we're studying it, but also humans have this gene. And dysfunction of this gene has been implicated in a variety of human disorders, as shown here in this wheel. And this includes metabolic disorders like obesity, diabetic retinopathy, and gout. And it includes things like cancers. And we're particularly interested in neurodevelopmental and neuropsychiatric disorders, which is one of the things that has really been shown to be associated with this gene when it's not functioning properly in humans. And so our goal is to find out what's happening in the brains of fruit flies, and then determine whether we can extrapolate that to what's happening in human brains. We've studied this gene in our lab for a while now, and this is from a paper from a former postdoc in our lab, and they found that when animals are stressed, there's a lot more of this RBFOX1 protein made. And when there's a lot more of the protein made, the protein is shown here with these little green circles, and it associates with a bunch of different organelles within the cell and can also form these fibers that are these aggregates of protein, similar to what I mentioned before in the case of tau. And the end result of this upregulation of this protein in a cell is to promote the cell's survival. And so when the cell is stressed, RBFOX1 can help it survive this stress. And now we've shifted to studying what happens specifically in the brain. And we're looking at brains in RBFOX1 mutants to find out what's happening in those brains. And so here are a couple of examples of some of the things that we've found. In the top row, I'm showing a control brain and a mutant brain. I think immediately you can probably see that there's a pretty striking difference there. And what we're looking at in green are the lineages of particular kind of neural stem cell. And a lineage is the stem cell itself plus all of the neurons that that stem cell has made. And you can see that there are a certain number of these. There are actually 8 of these clusters of neurons in each brain lobe. This is a brain lobe, and that's a brain lobe. And then what's interesting about this particular kind of neural stem cell is that these are the cells that make the central complex, which I briefly brushed past before, but it's a very important structure in the adult fruit fly brain that's responsible for navigation. And so now we want to find out whether this defect that we see in the RBFOX1 mutant, where some of those lineages are undetectable, we don't know if they've gone away or they never formed, we're not sure yet. But does this defect affect the formation of that structure in the adult brain? And then another phenotype that we're looking at, and this is from Sanjushri, a grad student in our lab. And she is looking at the mushroom bodies in the adult brain. The mushroom bodies are those L-shaped structures that I pointed out before that are the learning and memory centers in the adult brain. And she's found that in the RBFOX1 mutant, some parts of the structures are missing. So you can see that these L structures are quite perfectly intact in the normal brain, but then in the mutant brain, the part that's supposed to go up, the vertical part, is missing in some of these brains. And then we want to know a couple of things. We want to know why are these structures not forming correctly? And then furthermore, we want to find out, is there a deficit in the ability of these flies to learn? That's something that we have plans to look for in the future.
So in humans, so how does the mutations that you're studying in the fruit fly relate to what we know about the human version of this gene?
Yeah, good question. So what's known about the human protein is that it's a protein that binds RNA. And that happens in flies as well. It binds RNA. And when it binds RNA, it's instrumental in a couple of processes that happen in the cell. So when a gene gets turned on in a cell, the gene, which is DNA, gets turned into RNA, and then that RNA gets turned into protein. That's the typical central dogma that we learn about in genetics class. But when that RNA first gets made, it gets made kind of like a series of chapters in a book, introns and exons. And not all of those chapters are destined to end up in the final book. So some of the chapters get removed and some of them remain. But that can happen in a different way, so that not every book in the end ends up being the same book. They have different chapters. And what that equates to in a cell is that the final protein that gets made can be slightly different depending on which of those pieces got included in the protein. And RBFOX, when it sits in the nucleus, it is regulating which of those pieces get included in the RNA, which translates to what kind of protein gets made. And so that happens that's the most well-studied function of RBFOX1 in humans. And we know that also happens in flies. And so we're specifically looking at what happens when the protein is no longer able to bind RNA. Why does that cause defects in the neurons or in the structures in the brain?
Yeah, and I think that's an important point because if you think about a human brain, a lot of times you can't study these things unless the patient happens to donate their body to research after they pass away. Because I don't know about you, but I don't want somebody opening up my brain to take a piece out to look at these things happening inside a human brain. But because we have this protein that is found in a fruit fly and also found in a human and has a similar function, we can mess it up in a fruit fly and learn more about what types of changes that might be causing. And then we can eventually get to a point where we can do studies in other organisms and eventually potentially in humans to see if it's a similar process there. Any questions from you all? Yeah.
How similar or different was the anatomy of the fruit fly compared to, say, the ordinary vertebrate? For example, it doesn't have a liver, right? So you've got the liver disease up there associated with the RB-FOXP1, which is primary biliary cirrhosis. I haven't studied, so I'm
Yes.
curious.
So certainly one can't model everything in a vertebrate in a fly. And so we focus on the things that we can focus, that we can study, we can model. And the brain, I think, is a perfect example of that because there are a lot of similarities in how the brains work. But unfortunately, we can't model everything.
Yeah.
Has a gut for sure. Yeah. It has a gut and it has a kidney of sorts in that it has an excretory organ and can filter its hemolymph. It doesn't have a circulatory system the same way we do because insects have an open circulatory system. And instead of blood, they have hemolymph, but there is in fact a heart that's in the abdomen of the flies that pumps and circulates that hemolymph throughout the body cavity. And there are a lot of similarities in the cells that form that heart tube to cardiac cells. And many other examples of things we can model, but certainly there are limitations.
And I think that's an important point too, right? It's like the research never stops with just one organism or one lab. So the things that you find out about RB-FOX1 in a fruit fly, a researcher in a zebrafish lab or in a mouse lab or in another lab might be able to take that information and use it to form experiments and ideas that they can test that look more at how it might be doing a similar role in a liver or another organ that organism has. Because ultimately science builds on each other in that way. And a fruit fly is a great model in terms of the neurological system, but that information that this lab finds out could also be applicable to other organs and other organisms. And we won't know that until that research gets done. I saw another hand. Yeah.
Do you have a way to measure neuroplasticity?
The question is, do we have a way to measure neuroplasticity?
First, can you tell people what neuroplasticity is?
Neuroplasticity is how well a brain can form when subjected to some insult, like a mutation or an environmental problem that causes it to a trauma, something like this. So there are labs that study things like traumatic brain injuries in flies and determines
Neuroactivity, neuron transmission activity by age.
by age.
I mean,
In aged flies, you mean?
looking at the science of aging with humans.
Yeah.
This is of my interest, and it seems that there's some work being done, but I don't know where.
I think another thing here is neuroplasticity can also mean the changes in behavior of neurons after repetitive firing and as a component of learning and memory. and whether that might change with age.
Okay. Think how to answer this. There are certainly studies with aging, as well as looking at flies, young adult flies, where they do look at the function of the neurons themselves. You can do things like calcium imaging to see how rapidly neurons are firing, and whether that changes with certain treatments and things like that. And I don't know specifically, but I am certain that somebody is probably looking at things like this with aging brains as well, and see how that changes.
Other questions? Yeah.
In our body, the brain is separated from the circulating blood by the blood-brain barrier.
Yeah.
Would the fly have something similar?
The question is, do flies have a blood-brain barrier similar to what mammals do that separates the brain from the circulating blood? And the answer is yes, they have a blood-brain barrier. It's a bit different from what we have, in that it's made of a very thin layer of glial cells. Glial cells are a type of neural cell that serves to isolate and protect and neurons. And the brain lobes that I showed you, you can't see it because they're very small, but there's a very thin layer of glia that surround those brain lobes. And the important thing about a blood-brain barrier is that the cells that form that and protect the brain have to be very tightly attached to one another. And this happens through a junction called septate junctions that in mammals, these are tight junctions, and they certainly do have this.
For bonding in that layer.
An electrical charge for binding?
Yeah, a positive, negative, but you know.
Not that I know of, no. I've never heard something like this. It's really just, it's a very mechanical, the junction?
Yeah.
Yes, I mean, it's a very tight cell-to-cell junction that forms between these glial cells.
Okay, we're almost out of time. So I have a last question for you. What is something that you wish the public knew about your work?
Oh. So what I wish that the public knew, that I hope that this room knows now, is that even though they look so different, there is a great deal that we can learn from fruit flies. There's a great deal we have already learned from fruit flies about how their genes work, about how cells work, about how organs and tissues interact with each other. And with every generation, there are even more questions that come up and more that we can learn. And so, and not only that, a lot of what we learn from studying fruit flies is actually relevant to humans. And just like we were talking about how neurons operate very similarly in them and in us. So, and this is what I wish people had a better appreciation.
Any questions on the Zoom? We have one question here, and then do you have a question? Yeah.
Just thinking about the RB mutant, you're looking just at the brain, and you know that the gene is involved in many things. Have you looked at other organs or material in fruit flies and in the mutant, the RB1 mutant?
Yes, we have. So the question is, have we looked at other organs beside the brain in these RBFOX1 mutants? And yeah, we have looked at several. So the paper that I mentioned that was published previously, she looked in the ovaries of fruit flies, excuse me, as well as in cell cultures. And I have looked at testes of fruit flies, and we've also looked at, Sanju, what else have we looked at? Okay, so we've looked at other organs. what's interesting is that every place we look, we seem to find a different phenotype, and some new defect. And so in the ovaries, it's a great example. The ovaries, the germline cells, these are the cells that are going to go on to develop into an egg. And in the RBFOX1 mutant, that process cannot take place. They don't progress, they don't mature into an egg. And then also in the ovaries, a different RBFOX1 mutant has another phenotype that is in the cells that support the germline. And there are just too many of these cells. There are more generated when the fly is developing. And then in testes, there are also different phenotypes that we've looked at.
Jerry, you had a question?
Well, this is probably an unfair question, but I was on social media last week and I saw this announcement from the University of Maine alerting everyone to be on the lookout for the spotted wing invasive fruit fly.
Oh, yeah.
And this was news to me that I never even thought about.
Yes.
Kind of the danger that something like that can pose. I was just curious, how does something like that happen where you have sort of an invasive fruit fly that has different behavior kind of than regular fruit flies? They were saying it attacks ripening fruit instead of overripe fruit.
Exactly.
Do you have any thoughts about that?
It's a different species, has nothing to do with us. No, there are thousands of species of Drosophila fruit flies. What we study is one species, the species that Thomas Hunt Morgan started studying back in Columbia University. And it's Drosophila melanogaster, and it's quite innocuous. It's a pest in your kitchen for sure, but it's not a pest that is invading crops or causing devastation in your fruit yields and things like that. So the spotted wing fruit fly is a much bigger problem than if Drosophila melanogaster were to get loose.
So maybe we don't have an appreciation for those versions of fruit flies. But we are out of time. So one thing I want to leave you with is hopefully you now will have a different view of fruit flies. How many of you will think differently when you see a fruit fly in your kitchen next time? Anybody? Awesome. Well, thank you guys for coming, and thank you, Travis.
Thank you.
You guys, I think it definitely does give us a glimpse into the power of these comparative models that are utilized here at MDI-BL. You've heard a little bit about so many of them over the course of the summer, so it was fun to end our summer series with this one. So we have one more Science Cafe for you this summer, August 25th. We have Dr. Yannick Becker, who is here with us, who's going to be talking about the health of our vascular system. So we hope that you'll join us on the 25th to hear a bit about his work in sort of this protective mechanism in our vasculature and how important that is, and what maybe he'll give us some tips on how we can ensure that we continue to have that. So with that, we want to just thank everybody so much for joining us this evening, and hang around if you have questions. Enjoy some of the refreshments out there, and thank you all so much for being here. And thank you to Travis. That's great.
Thank you, Jerry.
Thank you. Awesome.